Good morning, good afternoon, and good evening. I'm delighted to welcome you to the latest BMKGENE webinar, how to study your first spatial transcriptome study. I'd like to thank you all for taking the time to attend today's webinar. Today's webinar will last for around 30 minutes, within 5 minutes for question and answers at the end. You could submit your questions via the Q&A button at the bottom of your screen, and we are looking forward to receiving your questions. And we will share the slides in a recording of the webinar via email after this webinar. Now I'd like to introduce the speakers of today's webinar, Lin Li, who's our Senior Product Specialist of Global Service Department. Lin, over to you.
Okay, thank you, Emma, and thank you so much for coming and joining us today. So today our topic is how to start your first spatial transcriptome sequencing study, and we'll introduce all the steps that involves in this project, from sample preparation all the way down to the biological insights. So before we move into our main topic, I would like to introduce myself again. My name is Lin, and I am now working as Senior Product Manager in Biomarker Technologies. We also call ourselves BMKGENE. So for some of you who may not very familiar with us, I'd like to briefly introduce Biomarker Technologies to you. So basically, we are biotech service providers, and what we want to do is mainly providing high quality, one-stop, high throughput sequencing services to our customers and help them to solve different research questions in terms of genomics, transcriptomics, and also metagenomics, and obviously single cell omics that I'm going to talk about today. Yeah, sorry. So I will just go strictly into our topic today, the spatial transcriptomics. So sorry. Okay, sorry, it's just a small technical problem here. Yeah, we have solved it. So in terms of spatial transcriptomics, when we look back into the RNA sequencing or transcriptome sequencing, so what next generation sequencing have brought us is that we can have a complete gene expression profile of one sample. So what we do is mainly we extract RNA from a sample or a part of tissue, and what we are doing at this step is actually taking an average of all the RNA expressions from every single cells in this tissue. So taking average is always a very risky thing to do in our research, obviously, especially when we are working with those tissues that with different cell types and they are very heterogeneous. So then that's where single cell mRNA sequencing come onto stage. By single cell RNA sequencing, we are able to cluster different cells with different expression patterns and align them with different cell annotations. But still, we are looking, still this is not enough for us, because in some cases, the spatial organization of different cell types is also very important to us. And that's why we are looking for new technologies or more and more technologies to help us to get the RNA expression profile, but at the same time can still retain the position information. So here I listed some common technologies on getting spatial transcriptomic data, and these are the ones that has already got commercialized. So the first one is region of interest, the RI, in which they mainly do the physical sectioning of your tissue, and you select the region that you are interested in and do RNA sequencing or microarray on top of that. And we also have SM-FISH and in situ sequencing, and these two technologies are mainly image-based spatial transcriptome study. And today, my focus is on the spatial barcoding technology, which means we capture the RNAs and barcode them on site, and then we harvest them from the site and do the sequencing to get the RNA expression profile and at the same time get their position information. So as you can see here, I put some images here showing that this spatial transcriptomic sequencing has been announced as method of the year by Nature Method. It happened like 1 or 2 years ago already. And you can see that during these days, the data that spatial transcriptomic data published along these years, like from 2019 until now, it got dramatically increased. And here you can see specifically for NGS barcoding based spatial transcriptomics, they accumulated a lot of data now. So this is some applications of spatial transcriptomics where that, as I mentioned, that when we are looking at some tissues that are very complex with very complex cell cluster organizations and etc. For example, immunology and oncology. For example, once we are looking at the tumor microenvironment, and if we are looking at the immunoinfiltration of the treatment of immunoinfiltration in these tumors. And also in the biological development studies, we are looking at the morphology related gene expression as well. That's where spatial transcriptomics can help. And in neuroscience, one of the biggest application here is to map these complex, say, brain structure in terms of cells and in terms of different expression profiles. And not only in human and animal or animal objects, it has a very large potential application in plant biology as well. For example, they're looking at the development of specific organs, for example, the roots, the stem tips, and etc. So this is where spatial transcriptomics really evolutionized our site in different topics. So in BMKGENE, we mainly provided services on this spatial barcoding based spatial transcriptomics. And one of the platforms we are using is 10x Visium that introduced by 10x Genomics. So here I showed a brief diagram on how it works. So basically we will have this slide that having 4 capturing areas on the slide, and each capturing area is around 6.5 by 6.5 millimeter in size. So what we will do is we mount our tissue slides on top of this capture area. And on this area, we will have these spots as I showed here. So on each spot, we will have different probes that can capture the RNA molecules released from the tissue slide. So these probes consist of these adapters that is compatible with Illumina sequencing flow cell, and we have spatial barcode where it retains the spatial information for us. And we also have UMI barcode, which means here is the molecule marker where thinking of like we have only a very limited amount of RNA released from one single spot. So we use this UMI barcode to help us to eliminate the bias or the errors during many rounds of PCRs and etc. And at the end, we have this poly-D tails where it helps us to capture the mRNA molecules by their poly-A tail. And once we have all these informations, we are able to transfer an image of this and combine that with their gene expression information and make more downstream analysis and generate a figure like this. So here it is. What it looks like the whole workflow looks like. And it seems a little bit complicated because there are so many steps. And here in BMKGENE, what we would like to do is to provide a one-stop spatial transcriptome sequencing service where we do all the steps in the middle that I showed here. And what we want from you is only your embedded OCT or embedded block or FFP block. And by the end, we will have a data analyzing report that contains all the information about the biological function interpretation and all the images, figures, and etc. So in the next several slides, what I would like to show you is that how it works from your side and all the way down to the data analyzing report that we will return to you. So first of all is the samples. So as I said, the samples that we used in spatial transcriptome sequencing is either OCT embedded blocks or FFPE tissue blocks. So for OCT embedding blocks, if you have already worked with, say, cryosectioning or other tissue-related or physiological studies, you probably have been very familiar with this. So this is very simple steps, simple experimental steps where you put OCT in this cassette and you have your sample ready and just dip it in and then do the freezing. They will form this block. And what we need is only this block. So there's one thing that I would like to emphasize that is that, as I mentioned, the capturing area of this slide is only 6.5 by 6.5. So the area that we can use, or the efficiency of these spots, is quite important as well. Here I listed some different occupation of this tissue on a slide. Normally we are expecting coverage of over 50% of the tissue over the capturing area. And also in the whole process, we will need like at least 3 blocks for this experiment. The first one is the block that we need to guarantee that the RNA molecule in the sample is still okay for the downstream analysis. So we will do a bulk RNA sequencing, bulk RNA extraction from the block 1. And once we are happy with the RNA quality, we'll move on to the real experiment with the second block, and we'll always have a block as a backup just in case something happens in the middle. So this is what we will do from our side. As I mentioned, the bulk mRNA QC, like already introduced, and then if that part is okay, what we will do is we will do a tissue sectioning and imaging on these blocks, and we will have a series of images generated, and we'll have a real-time video call with our customers to make sure that we are cutting in the right direction and we are harvesting the section that exact of your own interest. And what we'll do after that is that we'll do a permeabilization optimization. The reason for that is for different types of tissues or for different types of species, they will need different permeabilization conditions to make sure that they release most of the RNA molecules from the slice onto our capturing area. So for this permeabilization optimization step, what we will do is we will have a separate chip with 8 capturing areas. So the difference from this chip with the previous one is that they don't have barcode. They only have this poly-DT probes anchored on surface of this area. And once we try different permeabilized time for different slice, the RNA molecules will release from the tissue and then bind on the poly-DT probes on the slice, and they will have a fluorescent signal binded on top as well. And then for us to check whether the intensity of the fluorescent signal is good enough for us to say that this is the best permeabilization time for the specific tissue. And after that, we can move on to the real experiment where, like as I introduced, we mount the slides on a real capturing area with barcode, with UMI molecules and etc. And then once this cDNA synthesized from there, and we can harvest that and do the normal library preparation steps like cDNA amplification and library preparation. And here, once we have this library, we'll processed for sequencing. So now the main platform that we're used for sequencing is Illumina Nova 6000 and with library of P150. And in terms of the sequencing data, what we would recommend is that based on our own experience, like 50,000 P reads per spot is roughly a number of reads that we would like to generate to make sure that we uncover most of the genes that released from the tissues. And if we do a very simple calculation, say if we have 50% of tissue coverage, and that will generate like 2,500 spots, and then for each spot, we want 50,000 read pairs. And from there, we will generate around 250 million total reads. So, and there is another thing that I would like to emphasize is for different types of tissues, the RNA molecule richness is different. So for some tissues, they are more active, they will have more RNA expressed from their tissues, and then we probably need more data. It's mainly depending on the object that we are looking at. So here, normally we recommend to have roughly 30 to 60 gigabases clean data produced for each sample. And you can see for the sequencing reading part, the read one is mainly giving us the information on the spatial barcode and UMLIs that I mentioned. And the read two part is the one that read the exact insert sequence that we can map back onto the reference genome and get the gene information. And in data analyzing part, we have an integrated data analyzing workflow from raw data all the way down to the biological function annotation and enrichment. So here I would like to show you what you can expect from the data, the transcriptome, spatial transcriptome data analyzing part. So like all the other sequencing projects, the first thing is to make sure that the data is good enough for any downstream analysis. So this quality control points. So besides the basic quality control points for sequencing, like Q30, like read length, and etc., we will also need to check that if the accuracy on specific part is okay. For example, the Q30 basis in the barcoding region, which means we need an accurate location information, and the Q30 basis in RNA reads make sure that we are aligning we are getting the right gene information and aligning back on the reference genome. And the UMI is more on the accurate quantification of the expression. And besides the single base accuracy, we are also looking at this number, the median gene per spot, which means on each single spot, how many genes we can recover from our data. So normally for thinking of different tissue types and different species, we are looking for an average of 1,000 to 5,000 genes per spot. So the result, like the result I showed here, is pretty good, I would say. And once we are sure about the data quality and also the gene identification, we'll move on to the real analysis. So the first part is we can do data analyzing within one sample, which means we are focusing on the difference between different area within this one single tissue. So as I shown here, the first one is the gene detection intensity. The number of UMI more indicates the number of mRNA molecules we recovered from each single spot, and the number of genes, as mentioned, is it's a little bit different from the UMI because one gene can have many mRNA molecules that expressed. So here we can also have a heat map on this image on this HE image, which gives us a relative, like a rough idea of where it's more, say, active or where it is more stable in terms of the RNA expression. And besides that, what we will do is based on the RNA expression matrix on each single spot, we can do a cluster on each single spot. So it's like a PCA analysis where based on the gene expression profile, they can be clustered in different clusters and aligned back onto the tissue section. So for example, we know that in this image, this part is more or less similar. They share the same expression patterns, and they have completely different clusters here and etc. Another part in the inner sample analysis is the differential expression analysis. As I mentioned, like we would like to know what's the difference, like what are different within the samples. So on top of the clusters that we made in the previous step, what we can identify is those specifically high highly expressed genes in each single cluster. So in this heat map, we have each cluster line in these columns, and we have different genes in each line. So the red ones, for example, the red block here means these genes are the one that specifically high expressed in cluster 1, and they have others expressed differently in different clusters. And from here, we can also identify the marker genes or the marker genes of each single cluster. Normally, we regard the top 10 highly expressed genes in each cluster as marker gene of this cluster. And we can also have an expression intensity of each single gene on the slides as well. And here is another analysis on these hyperviable genes, which means these genes are jumping from different clusters, like they are either very extremely high expressed in one cluster, and they rarely express in the other. And these marker genes can be very important biomarkers, for example, in terms of marking different tissues, for example, normal tissues and disease tissues, and etc. So this is the analysis that we can do within a sample. And once we identify this like marker genes or highly expressed genes, we can align them into biological functions. These are the normal function analysis similar to the normal bulk RNA sequencing, like the geoclassification, protein-protein interaction networks, and KGG annotation, and etc. And also we can have this intergroup analysis as well. So this is the most commonly one, because normally we will have two different treatments, we have two time points and etc. We want to compare the difference and in more dimensionals. So here what we will do first is that we will combine all the spots of two samples together, or 2 or 3 samples together, and redo the clustering again. So in this case, they will have the spots from 2 samples, and once they reclustered, we will find that these when they align back on these slides, you will see like in the green arrow, they have these common clusters, but sitting on different positions for sure, because there's no strict repeats in terms of spatial, but you can still find these common clusters, and they also have these unique clusters here. So the differential expression analysis between different groups or between different samples can be done in different ways. So the first one is similar to what we did for the inner sample one, is that on these new clusters, we would also do this expression profile heat map, where we can identify the marker genes of each cluster. And also we have this expression profile in different samples as well. And they can all be aligned back to the different tissues. We can see, for example, some marker genes are highly expressed in this part, and in the other sample is in a different part. And what else we can do on this is that for those common clusters that presented in both tissues, we can also do this differential expression analysis to find, even they are common clusters, we can find the differential expressed genes. We can find these minor differences between them. And again, once we find some genes or gene patterns that we are interested, we can always align them onto function analysis. So these are the standard analysis we can perform from the transcriptome data. And what else we can do is a more customized data interpretation. For example, because you know the best of the tissues and you know where is the exact region of interest you want. So what we can do is we can draw a circle on the tissue, and we want to compare, like, for example, this part with another part, and this all can be done in the data analyzing part. So here, what I would like to emphasize is that during the spatial transcriptome sequencing analysis, I always said it's one spot or cluster of spots, because here the spot is not single cell. So normally it gets confused, but thinking of the size of the spot, for example, in 10x Visium, the size of the spot is around 55 micrometers and the resolution is around 100 micrometer. So within the resolution of that, it covers normally 4 to 5 cells. So this cannot give you an exact single cell resolution, or it cannot be perfectly aligned with a cell type. So normally what we do in this type of study is that we normally join the single cell RNA sequencing data with the spatial transcriptome data. What we can do is from single cell RNA sequencing, we can have this cell type annotation, and then the spatial transcriptome sequencing gives us the cell localization information. So basically the joint analysis is mainly on the correlation between different clusters, either different clusters generated from two different sets of data. For example, once we correlate the cell clusters with the spot clusters, we know that this part of the spots are more likely to be certain cell type, and then align the cell type on top of this position information. So as I mentioned, like in 10x Visium, we are not really looking at a single cell resolution here, but the development of spatial transcriptomics normally go into two directions. The first one is that number of genes that we can measure from one single spot. That gives us much more information within one spot. And the other direction is that we want to go higher resolution. We want to go not 100 micrometer, want to go higher, like 50 or even 5 or even single cell. And here I listed some technologies that is also spatial barcoding based technologies that released just these 2 years. And what they are doing is also trying to get higher resolution and at the same time get more genes detected within a single area spot. So here I would like to introduce that in biomarker technologies Beijing, we launched S1000 subcellular level spatial omics chip, and it just released 2 months ago in April. So here what we achieved is that we can achieve the resolution as high as 10 micrometers, so 5 micrometers. And what else we can do? Sorry, we probably, yeah. So here is an image from our R&D project. From R&D project. So if we zoom in this single spot, we can see under each spot here, under one cell, like I squared out here, there are 4 to 5 spots that covered by one single cell, which means we are reaching a subcellular level. But we are not really looking into subcellular level directly. What we can also do is that it can be customized. We can looking at resolution at different levels. For example, we can cluster several spots together to become a super spot and to achieve levels from 5 micrometer all the way to 100 micrometer. And with this type of chip, we also developed our own tool and data analyzing toolkits named BST Matrix. So what it can do is it can perform these data analyzing from sequencing data all the way down to a web report that just like the things that I show you in the previous slides. And it contains these 5 different small tools, but FASTQ to BC UMI, and also UMI to genes, which gives you the annotation of each molecule, and also the matrix make, where they generate the gene expression matrix for every single spot. And also on this toolkit, as I mentioned, what it can do is that you can adjust the resolution level as you want. So for example, on this tissue, you can either looking at it at 100 micrometer. If you don't want to look too much details, you can set it as 100 micrometer in terms of resolution. It gives you roughly 3 clusters, spot clusters. And if you go lower, you will have more complex structure. And if you zoom in, you can see there are 1, 2, 3, 4, 5 different clusters when you're zooming into, this is, I think this is around 20 micrometer. Yeah. So here it is more clear case on why we want to go for higher resolution. So this is a HE staining image of a hippocampus, and you can see on this HE staining there is a clear structure on these staining images. And once we are looking at the general cluster or general tissue organization, we can see at 100 micrometer resolution or 50 micrometer resolution, the general cluster organization is kind of more or less similar. But if we are looking into more details on, for example, the edges or on these small regions, what we find is that once we increase the resolution from 100 to 50, even to 35, they start to form more and more clear structures that can be better aligned back to this HE staining. For example, here, there is only one spot layer shown on this 100 micrometer data, but you can see on this HE staining part, you probably see this small empty part here. This is like a different cell type or different type of tissue, and it can be resolved on this 35 micrometer resolution. And in 20 micrometer resolution, thinking of the limitation of the detection, maybe there are a bit more noises, but it can still gives you a very clear structure of this tiny organization. So in BMK, we have accumulated a lot of experience on different tissue types and different sample types. I listed here, I list some of them here. So most of them are human samples like embryos, like kidney or tumors and etc. We have also processed a lot of tissue types from mouse or rats, and also we have experience in plants spatial transcriptomics as well, and there are more ongoing. And so at the end, I would like to briefly introduce biomarker technologies to you. So although we are rather new to our European customers, but we have already provided high quality sequencing services for Chinese customers for over 13 years. And our headquarters is in Beijing, China, and we have 2,000 square meter lab space in China as well, in mainland China, in Qingdao. And we have recently just launched our automated liquid workstation where we can achieve this automated library preparation steps and sequencing center. And also we have another bioinformatics centers seated in Wuhan, China as well. And this new lab in Münster, Germany is just launched 1 or 2 months ago, and hopefully we can provide more or better services for our customers in Europe from there. So besides the spatial transcriptomic services that I introduced in the previous slides, we also provide sequencing services for all different research arenas, for example, genomics and population genetics, transcriptomics, microbial genomics, and also we have third generation sequencing platforms as well. And for delivering high quality sequencing services, we are backed up with a very high, we are backed up with over 500 professional staffs in Beijing and in Germany. And also we have over 60 nation patent of inventions, and we have our own software copyrights of 200 of them. And we have with help of all these platforms and people, we have achieved a lot of, we have already contributed in a lot of high impact studies, and they are published in many journals as I listed. So this is, I think this is all that I would like to share today. And thank you so much. I think I get some questions here. Yeah, and if you have questions, you can leave a message in our Q&A session as well. So So one question from Anna on which plant tissues has this type of analysis already been applied to, only leaves or roots? So most of the, in the plants projects, we have done leaves. So leaves is, but leaves sometimes is limited by the thickness of the tissue. And we have done some roots, but it's like more fresh roots, like the tip of the roots is more preferable in this plants experiment. We have also done a flower, and yeah, I think I can move back on this. Yeah, this one. Yeah, we have done stem and flower, and some of the roots, like the fresh roots and germ and grain. Yeah, these are the experience that we have. I get another question is how many genes can be identified per spot? So yeah, so on each spot, so normally we are looking for 1,000 to 5,000 genes per spot, and it also mainly depending on the tissue type. So for example, your tissue is activate and you have more RNA expressed and it's more rich in these gene expressions, we probably will get more genes identified per spot, like say 6,000, 8,000, or sometimes even 10,000. Yeah, but for some tissues, probably they are not very active and we have like 2,000 or 3,000 per spot. Okay. Yeah.
Any other questions? Okay, thank you for you all attending our webinar today. And if you have any questions, you could also email us. Or okay, we got a new questions.
So I have a question regarding the combination of single cell sequencing and spatial transcriptome sequencing. Do we get the both in one-step procedure? No, actually, this is two different projects. So in single cell sequencing, what you need to do is you need to dissociate your tissue into single cell suspension. And we will do that on a separate system called 10x Genomics Chrome, where they form this germ and they capture each single cell and barcode it individually. And that's how we get we align RNA sequencing information back onto each single cell. And then from that data set, with the spatial transcriptome data set that I introduced today, we can align these two cell clusters and spot clusters, and then give a cell annotation on the slice. Like if you still remember the image that I showed. Yeah. Okay. Okay, so and the high resolution leads to more gene reads, or rather just mapping the location of in certain space. It's a very good question, because some of people may think that with higher resolution, probably we need much higher or much higher needs in the data production, but actually thinking of they have a certain number of mRNA molecule expressed in your tissue. So what we need to do is just capture most or capture all the mRNAs released from the sample and then barcode them with UMIs. So it's not necessarily to generate more data for a higher resolution, but actually we are using same, it's more like we have same number of reads generated for a more detailed clustering of these data. So to answer your question, with a high resolution cheap or high resolution study, we actually just need the same or like similar amount of data produced. Yeah. Okay. So I think that's all the questions that I get today. And as Emma said, we will have this recorded and we can send you this on-demand webinar video link as well via email. And if you have any other questions or if you want to start your own spatial transcriptomic project, you're always welcome to drop us an email or visit our website and just type in these forms and and send us any messages, and we are more than happy to help you to start your first spatial transcriptome sequencing project. And thank you so much again for coming and joining us today, and have a nice day.